about treatment strategies for deficiency of adenosine deaminase 2. Definition. Deficiency of ADA2 data is the first molecularly described monogenic vasculitis syndrome. DADA2 is caused by allelic hypomorphic mutations in the ADA2 gene that encodes the Adenosine Deaminase 2. Pertaining vasculitis and inflammation can affect many organs, explaining the intestinal hepatology.
I think the transformative potential of gene therapy is that it is a one-time treatment which has the potential to provide a long-lasting benefit because it goes at the root of the disease. If the disease has a genetic cause, we can treat the genetic cause by replacing the gene.
The marketing approval for the SCID-ADA gene therapy represents for us fulfillment of a promise. We cannot be satisfied if we only fund outstanding, excellent research. This is the mean, but the objective, the final objective, is to find it.
However, often with flares and inflammation and vasculitis often separate as cytophrine, cyclosporine, tacrolimus, and cephalosphosphamide amyloidexam.
SCID. SCID stands for Severe Combined Immunodeficiency and is a catastrophic disease in which patients have no immune system, the bubble boy syndrome as it's commonly known. This was an attempt to develop a new form of therapy that would be more effective than prior attempts, that would be safer, and that would be available to older children who have previously failed a standard bone marrow transplant. We saw very high levels.
Two pros and two cons. The treatment with biologic TNA inhibitors was successful in controlling the fever episodes, vasculopathy, and prevention of strokes. In outpatient reporting, online, in standard care for stroke patients, it is recommended to discontinue treatment with acetylsalicylate.
It takes your lectures and notes to create a personalized study plan with exclusive videos, practice questions, and flashcards, and so much more. Try it free today. Lesch-Nyhan syndrome is a rare genetic condition which leads to excess uric acid in the blood, and it causes kidney, joint, neurological, and behavioral problems. Normally, each cell contains both DNA and RNA. In diving deeper, each nucleotide can be broken down into a sugar, a deoxyribose in DNA or a ribose in RNA, 1 to 3 phosphate groups, and a nucleobase, which can be either a pyrimidine or a purine. There are 3 pyrimidine bases, cytosine, thymine, and uracil, and 2 purine bases, adenine and guanine. So the name of a ribose-containing monophosphatic nucleotide based on guanine would be guanosine monophosphate, or GMP for short, whereas one based on adenine would be adenosine monophosphate, or AMP for short. Now, there are 2 ways our cells can make nucleotides. One is from scratch, also known as de novo synthesis, and the other is the salvage pathway, which recycles nucleotides that are already semi-degraded.
Issue applied in Ecuador. In Ecuador, there are few epidemiological data on the prevalence of this syndrome's treatment.
Those children affected are missing a protein, Adenosine Deaminase, fundamental in producing lymphocytes, cells which are responsible for defending us against infective agents such as viruses, bacteria, or funguses. In normal conditions, the lymphocytes are formed from the stem cells inside the bone marrow and, once freed into the bloodstream, recognize and neutralize the infective agents. Without lymphocytes, viruses and bacteria freely attack the organism. Even banal infections may become so dangerous as to force these children.
Conclusion. The main state of treatment is TNA inhibition, which is cell in suppressing inflammation and in prevention of vascular events. HSCT may be an option in patients presenting with hematological disease and immunodeficiency.
Treated with an ADA skid disease, Strimvelin. Emily Kalm-Sema, working in the Gene Therapy Rare Diseases Unit at GSK, company behind Strimvelin, is here with us today. So Emily, tell me about GSK's history and its involvement in gene therapy.
Yeah, fantastic. Well, GSK in the Rare Diseases Unit has had quite a long history in cell and gene therapy, actually. We have enjoyed a fantastic collaboration with the scientists and clinicians at the Telethon Institute for Gene Therapy since 2010. So that's about 7 years now.